Okay. If we want to plug inbreeding in to Hardy-Weinberg equations, we can define alleles in two different individuals as identical by descent if they descend from a recent common ancestor. Okay. Quick example. Eye color in humans is actually polygenic. It's governed by multiple genes, but roughly brown is dominant to blue. I've got brown eyes. My sister has brown eyes. And I believe the Emperor of Japan has brown eyes. Okay, so we've all three of us have alleles for brown eye color. My sister and I share alleles by recent common ancestry, right? Because we both inherited those from our dad. My mom's got light eyes, so presumably we both got brown eye genes from our father. Now, I have no known connection to the royal family of Japan. Now, ultimately, I do if you go back far enough. I think someone worked out that any two people on the planet are at least 56 cousins of each other. You know, everybody shares common ancestry if you go back many thousands of years. But I certainly don't have any recent common ancestry with the royal family of Japan. So in that case, my brown-eyed alleles and the emperor's brown-eyed alleles would not be identical by descent. Half a tick. I think I went forward a slide without intending to. Yeah, so my brown-eyed alleles and my sister's brown-eyed alleles are identical by descent because they both come from my father. My brown-eyed alleles and the emperor's brown-eyed alleles would not be identical by descent because they don't come from a recent common ancestor. And in any given population, the fraction of alleles that are identical by descent is capital F, and we call that the inbreeding coefficient. Think back to the last, was it Monday? When I was having you imagine that oyster bed with all of those oysters that are firing off big clouds of eggs and sperm into the water and letting the water currents do the rest. Well, the probability of, okay, yeah, the fraction of alleles in that population that are identical by descent and would be F. And if I'm randomly pulling gametes out of a pool, the probability of me pulling out, let's say, a capital A egg is P, if we let P be the frequency of big A alleles. Then the probability of me pulling out an egg that's got the big A allele is P. The probability of my pulling out a sperm that's identical by descent to the one that the egg already has is capital F. So the probability of me pulling out an egg with big A is F. Start over. The probability that I'll reach into that gamete soup and pull out an egg that's got big A is P. The probability that I'll reach in and pull out a sperm with alleles that are identical to the egg by descent because they come from a close relative of the female that made the egg, that probability is F. So the probability of getting a big A zygote with alleles, big A alleles identical by descent is PF. The probability of getting a big A zygote with alleles that are not identical by descent is P times 1 minus F. That's the probability of randomly pulling out an egg with big A times the probability of randomly pulling out a sperm with big A times the probability that that sperm is not identical by descent to the egg. P times 1 minus F. We can show the probability of getting a little a zygote with alleles that are not identical by descent is Q squared 1 minus F. The probability of getting a heterozygote is 2pq times 1 minus F. We can do some rearranging of all of this, and in the interest of time, I'm not going to work through all of this in terrible detail. We're coming up with a simple, this is me deriving what's going to be a fairly simple formula. In a population where we're looking at inbreeding, the total frequency of big A genotypes is the sum of those two previous formulas. It's PF plus P squared 1 minus F. And we can rearrange that to P squared plus PQF. So think about this for just a minute. If there's no significant inbreeding in our population, and there's no selection or anything else going wrong, then the frequency of big A homozygotes would just be P squared. Yes? Right? Okay. If there is inbreeding going on, then the frequency of big A homozygotes is going to be P squared plus something. That sets up the telltale sign that inbreeding is going on in a population is the frequency of homozygotes is higher than it should be, and the frequency of heterozygotes becomes lower. Okay, and in fact, if inbreeding is going on, if there's no inbreeding going on, then F is 0, and that PQF just drops out, and we're in Hardy-Weinberg land again. If inbreeding is going on, the frequency of big A individuals is P squared plus PQF. The frequency of little a genotypes is going to be Q squared plus PQF. And the frequency of heterozygotes is 2pq multiplied by 1 minus F. Okay, we're not at the important part yet. I think this is the important part. Okay, in a population, we can go in and just count the number of heterozygotes. And we can abbreviate that H sub 0. Heterozygotes observed, H0. Or not H0, it's HO. H subscript O. So if you are in a population where inbreeding is going on, the observed frequency of heterozygotes is going to be 2pq times 1 minus F. And we can solve that for F, and we get F is equal to 1 minus the observed frequency of heterozygotes over 2pq. 2pq is the frequency of heterozygotes we'd expect if there's no inbreeding. And we can just call that H sub E. So what is F? This is the equation you need to know. F is equal to 1 minus observed heterozygotes over expected heterozygotes. That's what you need to know. If there isn't any inbreeding going on and we are in Hardy-Weinberg land, then the observed frequency of heterozygotes should be the same as the expected frequency of the heterozygotes, right? Yes, you with me there? If there's no inbreeding and we're meeting all of the Hardy-Weinberg conditions, then observed heterozygotes should equal expected heterozygotes. And HO divided by HE would be 1, and F is 1 minus 1, which is 0. If there is inbreeding going on, then we're going to have a deficit of heterozygotes. And observed heterozygotes divided by expected heterozygotes is going to be smaller than 1. And F will be 1 minus something smaller than 1, which will be a positive number. Okay, I'm not spending an awful lot of time on the derivation. You can go back and look at the slides if you want to. If you just want to see what the formula looks like, this is one I would expect you to know. that the inbreeding coefficient is 1 minus observed heterozygotes over expected heterozygotes.
So, wait, so F is going to be positive.
Yes. I mean, it could be 0. If there's no inbreeding going on at all, then F will be 0.
If there is inbreeding, F
Mhm.
will be positive.
Yeah, it is. No, wait. You could get if you have more heterozygotes than you would expect, then HO over HE would be greater than 1. And yeah, you can actually get a negative F.
And,
Okay, I take that back. You can get a negative F sometimes.
but it doesn't mean that it's inbreeding. It's.
Okay, excess heterozygotes would actually be, that's another violation of the random mating criterion. And like if the critters in your population were deliberately mating in such a way that they're producing lots of heterozygotes, that can happen. Okay, there was an experiment done with college students years ago. Very popular psychology experimental subjects. They took some men, had the men wear t-shirts for 2 days, and then put the t-shirts in clean cardboard boxes, presented them to college women, and had them sniff the t-shirts. And in some cases it probably smelled horrible, and in some cases, hey, there's kind of something there. And they asked them to rate the attractiveness of the aroma. The other thing they did was they tested everybody's genes, and they found out that women were likelier to rate as attractive the aroma of men that had different alleles from them. And I know smell is not the only thing by which we choose partners, but it may be more important than we think. And it seems like there is, to some extent, at least as far as smell goes, women have a tendency to be attracted to men that are genetically different from themselves. If something like that is going on, where individuals in a population are likelier to mate if they're genetically different, then you have kind of the opposite of inbreeding. And you could actually end up with a negative F that way, because a mating system like that, if all of the big A's in the population really want to get with the little a's, and vice versa, you'd end up with more heterozygotes than you would expect. Now you'd end up with deliberate outbreeding. That's a violation of the random mating criterion too, but it's a violation in the opposite direction. So you can end up with a negative F. All right. So inbreeding increases homozygosity in a population, and here's where this gets into practical applications. Traditionally, animal and plant breeders use this to fix desired traits. If you're developing a new breed of, if you're breeding racehorses, all right, you do not want to cross 2 racehorses together and get a pony. You do not want to cross 2 racehorses together and get a Clydesdale, one of the really big horses that used to be used for pulling carts or something like that. If you're breeding racehorses, you want to cross 2 racehorses together and get another freaking racehorse. Yes, right? If you're an animal breeder, you don't want genetic variability. You want all of your individuals to be the same. If you're breeding plants, you want to breed, let's say, a variety of tomato where all of the tomatoes can be harvested the same way and used in the same way, right? You don't want to cross 2 cherry tomatoes together and get a Cherokee purple or something like that. Yes? If you're a commercial breeder, you don't want variation. So what animal and plant breeders have traditionally done is inbreed. Because if you inbreed a population, you get lots of homozygosity. And if you couple that with selection against traits that you don't want, you'll end up with a population that is homozygous for all of the genes that you want it to have. You want to have a farm full of racehorses where all of the horses are homozygous for all of the genes that make the things run fast. Yes, right? So if you're an animal breeder, you want standardization, and the way you do that is by breeding close relatives. It's not uncommon, people developing an animal breed to breed siblings or to breed father to daughters or something like that. The problem is that that increases homozygosity for traits that you want. It also increases homozygosity for traits that you don't want. That's a Doberman Pinscher. Had a professor in grad school who loved Doberman Pinschers, said they were the best. They're loyal, they're playful, they're good guard dogs. They're what's that?
Feisty.
They're feisty. Okay, but yeah, he said they were terrific. And the thing about Dobermans is that, like most dog breeds, they were developed from a very limited number of founder dogs that have been inbred among themselves for generations. So Dobies are homozygous for all of the things you want in a Doberman Pinscher, namely looking like that. But there's also a high incidence of a bleeding disorder called von Willebrand's disease. Dobies are prone to a particular type of hemophilia because they're inbred. And there's a high number of Dobies that are homozygous for the VWD gene. You get that a lot in like Dalmatians, right? Dalmatians have been very carefully bred to have spots and look cute and have lots and lots and lots of energy. Dalmatians were originally bred to be dogs that would run beside your carriage. That's why they're associated with fire, by the way. Firehouses would have Dalmatians to run beside the fire truck when the fire truck was drawn by horses back in the old days. Right? Even now, though we don't need them now, people still think of Dalmatians as being fire dogs. Right? Like Marshall on Paw Patrol, of course, he's a Dalmatian. Sorry, my kid is 11, and when he was 3 through 6, we watched a lot of Paw Patrol. Oh God, did we watch a lot of Paw Patrol? And that's why Marshall is a Dalmatian, because Dalmatians are associated with fire, because Dalmatians were bred to run beside your carriage, and that's why Dalmatians are so energetic. You should not keep a Dalmatian if you're not going to let it run for miles every day.
Aggressive.
They're aggressive as well. They're trying to keep people back from your carriage. They're also very highly prone to deafness. And they've been inbred for all of the traits that people want, for spots and high energy and fast running speed and willingness to bite people to keep them from messing around with your carriage. But inbreeding also means they've got high levels of homozygosity for deafness alleles. And registered Dalmatian breeders are supposed to euthanize any dog that's deaf. They're supposed to do that even if they can find people who wouldn't mind taking care of a deaf Dalmatian. Dalmatian breeders are trying to select against deafness in pretty much the most direct way possible. Maybe it would be nice if they didn't, but that's what they do. So here's another example. Well, this is part of the reason why an awful lot of dog breeds have tendencies towards diseases. Boxers tend to be prone to cancer for some reason.
Weenie dogs would have back problems.
Right, although in some cases that's not so much because they're homozygous for a gene for back problems, but they are homozygous for genes that give them short legs that can't absorb a lot of shock. So they'll hurt their backs if they jump off the sofa too often.
Bulldogs have breathing problems.
Yeah, so do pugs. Yeah. But sometimes you'll get breeds that have a high incidence of a disease that's not obviously related to their physical features. Like nothing about a Doberman suggests that they ought to have a high risk of hemophilia, but they do. And it's just because the alleles for hemophilia have, because that's an inbred population, you get a lot of dogs that are homozygous for hemophilia. And yeah, here's another example. That is, of course, a white tiger, not actually albino. An albino would not have any black stripes. This one, the white tiger is not producing the orange-brown form of melanin, but it is producing the black form. That's technically called being leucistic. It's different from albino. The last wild-caught white tiger was captured in India in 1951 and was named Mohan. And the rich guy that owned Mohan, well, he bred Mohan with an orange female named Begum up there. That's their family tree. And Mohan and Begum only had orange cubs. Those are represented here by these circles with a slash through them. Those are heterozygotes. They're orange, but they're carriers of the leucistic allele. So Mohan got bred to his daughter, Radha, right there. And Mohan and Radha had some cubs. There's more off the screen. I didn't have room to show the entire pedigree. But there's a litter of cubs that Mohan and Radha had. And they include Sakeshi, Mohini, Rani, and Raja. And Mohan had cubs with his granddaughter, Sakeshi, right here. So Mohan was bred to his daughter and his granddaughter. And that gives you homozygous white tigers in the offspring. And all of these down here at the bottom, Gomti and Gautam and Govind and Lakshmi and so on, those are all white tigers. And that lineage has been maintained by zoos and breeders ever since then. I don't know if you folks remember Siegfried and Roy. There were a couple of magicians in Las Vegas that had this act with a lot of wild animals in it. They do magic illusions with live lions and tigers and things. And then one of them nearly got killed. Kind of a risk when you're involving tigers in your stage show. And yeah, they haven't performed since then. But they used to have white tigers on display a lot. And this is this, beautiful exotic thing. What's that? Nope. No, the white tigers have only been kept in zoos and by private tiger breeders. Although not that bitch Carol Baskin. Anyway, the problem is that Mohan's descendants, being descended from Mohan mating with his daughter and again with his granddaughter, they're homozygous for white fur. And if you breed two white tigers, you'll only get white tigers because they're homozygous for those alleles. But a growing number are homozygous for other recessive alleles that you don't want. So that's a white tiger with skull malformations that mean that he can't close his mouth. Okay, because when you inbreed, you get a population that might be homozygous for what you want. It's also homozygous for what you don't want. Now, in the old days, you would inbreed, and the individuals that were homozygous for what you wanted, you keep, and the individuals homozygous for what you didn't want, you euthanize. So if we really wanted to, we could create a breed of white tigers by inbreeding them, picking out the best white tigers, but putting down all of the tigers that showed any traits that weren't desirable. But in fact, it hardly seems worth it. The Association of Zoos and Aquaria, this is why they banned its members from breeding white tigers in 2011. I mean, I'm sure there are still private breeders and zoos that are outside the AZA that will continue to do it. But accredited zoos, zoos with a good reputation no longer breed white tigers. just because it produces too many offspring that have health problems like this poor guy does. Okay, well, the nuke should be inbound in a few minutes. Right. Sometimes, on the other hand, even that disease tendency is what we want. There is a huge lab in this country called the Jackson Laboratories, jacksmice.jax.org, that has been maintaining strains of inbred mice. This is one of them. These are mice from their catalog. And if you do like biomedical research or cell biology or neuroscience on mice, you're going to work with the Jackson Lab, because they maintain spreads of mice that have been inbred for at least 20 generations and sometimes hundreds. These strains that have been breeding brother to sister for all this time. And that makes these mice homozygous at practically all loci. There's no genetic variation left in this population. They're all homozygous at virtually all genes. There's no genetic variation. The reason why you would actually want to do this to these poor mice is it creates a pool of mice whose genes are all the same. Now, you could do cloning, but cloning is expensive and frequently doesn't work and difficult and has some other practical drawbacks. Instead of cloning, you could just inbreed your mice brother to sister for 20 generations, and you end up with almost the same thing. A bunch of mice that are virtually all the same. And now you can do experiments on these inbred mouse lines without introducing the variable of genetic variation. Right? If you caught a bunch of wild mice and you all gave them a drug, if they're all genetically different, they're going to vary in how they respond to the drug, right? If your mice are practically Xerox copies of each other, they should all respond to the drug in the same way. And in fact, many of those strains are homozygous for genes that mimic human disease. That's a mouse from a strain, I don't know how you pronounce that, NOD/SHILTJ. These are Nodshit mice, or Nodshilts. Nodshiltjes. Okay, these things right there, if you feed them an appropriate diet, they're guaranteed to develop insulin-dependent diabetes. Obviously, this is not very good from the point of view of the mice, but it's handy for diabetes researchers because you can have this pool of experimental animals that are all virtually guaranteed to develop insulin-dependent diabetes, and then you can start testing, drugs and other treatments on them. There are strains of inbred mice that are virtually guaranteed to get cancer. I think you still have to expose them to a carcinogen to be sure. But these inbred mice, if you shave a patch of skin on their back and you paint it with a certain chemical, you're practically 100% of the mice will get tumors. There are mouse models for coronary obesity. There are mice that are pretty much guaranteed to get obese if you feed them a particular diet. We used to have a researcher here a long time ago who was working on OCD, and I think she had some inbred strain of mice that had a behavior pattern, like compulsive, I think it was compulsive mouth movements, something like that, that was considered to be an appropriate experimental model for OCD. She's not here now, and the reason is that the other thing she spent a lot of time studying was the effect of cocaine on the human nervous system. Yeah, specifically hers. And that's the thing, she came from a pretty rich family because most of your faculty at UCA couldn't freaking afford a cocaine habit if you wanted to. They don't pay us that much. Harvard, sure, but
UCA.
UCA, we are pretty much kept honest. I mean, except for cooking meth, but, what's that? Arkansas has the average lowest faculty salaries in the South, and the South has average lowest faculty salaries in the country.
Yeah, we're a poor state.
What's that?
I said we've always been a poor state.
Yeah, pretty much. Oh, well. Oh, one last thing. Are we almost out of time? Okay, it is? Okay, can I do this real quick? You can take two inbred lineages and cross them. And first of all, not only will the offspring be heterozygous at almost all loci, so you could cross a mouse that was inbred to have diabetes with a mouse that was inbred to have OCD, and the offspring being heterozygous, you would expect would have neither. But often the offspring, aside from that, they're often larger and healthier than either parent, and we call this heterosis, aka hybrid vigor. So there you've got two different inbred strains of corn, and then the F1s in between are taller plants with bigger ears. And this was published in 1909. They do this with a lot of crops. Grape tomatoes are relatively new. They came on the market around 97. About the time I came here to teach. I don't remember seeing these as a kid. And they're actually a hybrid. In the trade, they're known as Santa F1. And so you get grape tomatoes by crossing two different tomato plants and planting the seeds of the cross. And exactly what those strains are is a trade secret. You don't want anybody to be able to grow grape tomatoes. There's one company that's got a monopoly on the knowledge of what two strains you have to crossbreed in order to get hybrid grape tomatoes. Farmers always used to save a portion of each year's crop as the seed for the next year's crop. So there you've got a corn farmer who's being told to save some of his corn and not sell it and use the seeds to plant again next year. There are still people that do this, and it's actually popular in the vegetable gardening community. There have been varieties of vegetable that have been maintained in home gardens for hundreds of years just by saving seed. You save some seeds from your tomatoes one year, and then you plant them the next year, and you don't have to go out and buy more tomato seed. Large-scale farmers don't do this because beginning in the 1930s in corn, farmers have been encouraged to purchase hybrid corn seed. So there you've got, this corn is so healthy that it's now the golden snitch. Like, look at it, it's got wings, and I fully expect to see a bunch of farmers on, Iowa farmers wouldn't ride broomsticks, so they'd be riding pitchforks trying to catch the golden DeKalb hybrid quality corn snitch. What the hell was that? Okay. That hybrid corn was vigorous, it's got this heterosis. It often blends desirable traits from two different strains. Like you could breed an inbred corn that was big with inbred corn that was disease resistant, and you get hybrid corn that was both. And this is a large part of why in 1930, we were producing, that's tons per hectare. We went from about 5 tons per hectare to, in 2010, nearly 9 to 10. We've nearly doubled crop yield since 1930. The price is that farmers have to buy their hybrid corn seed from seed companies because hybrid corn seed isn't going to breed true. If you get hybrid seed, plant it, harvest the corn, and then save some of those kernels to plant next year, you can get anything. You're going to get all sorts of trait combinations, many of which are not going to be worthwhile. So at a small price paid in independence, hybrid corn has greatly increased total yield. And there, I'm done. All right, sorry we went a little bit long, but I did want to cover this and maybe bring it back to some practical, real-world concerns. Hang on, and stop recording.